A warm welcome to everyone. I welcome you all to this animated video. Let's get into the video. Glycogenolysis is a process of breaking down glycogen into glucose and produce energy to maintain the blood glucose levels. If we look at the word glycogenolysis, it is telling us what does that mean. Glycogen comes from a polymer glycogen that is made up of glucose, and lysis means to breaking or killing it. So in this process, we are basically going to break down this glycogen in order to get glucose from it. In normal circumstances, this glucose is usually available to us by food that we eat, specifically the carbohydrates that we eat. But if we take too much of that carbohydrate inside our body, then that excess amount of glucose will convert itself into glycogen, and this process is called glycogenesis. At first, glucose is converted into glycogen, and this process is called glycogenesis. And the reverse of this process, where the glycogen is converted into glucose, is called as glycogenolysis. When does this process called the glycogenolysis occurs? This occurs when there is a low blood glucose. The low blood glucose is also termed as hypoglycemia. The main hormones that stimulate the process of glycogenolysis are glucagon, epinephrine and norepinephrine, where glucagon comes from the liver and epinephrine and norepinephrine are derived from muscle and as well as liver.
The excess amount of glycogen will deposit itself in liver, in muscle cells and in brain cells, specifically astrocytes. The other organelles that can also deposit this glycogen are heart, kidney and fat cells. Well, if too much glycogen deposits inside our fat cells, that could be a problem for us. That's why the time when we sleep and people who fast and those who exercise, all these things give the opportunity to our body to convert this deposited glycogen back into glucose.
Now, the cells that are going to change the glycogen to glucose have two different types of targets. The first type of target is to gain instant energy, for example, like heart, muscle, and liver cells. So, if our blood is having low glucose, then the liver cells going to convert the glycogen to glucose. Inside our blood, we have two different types of options. One, the low blood glucose level, and a high blood glucose level.
Inside the blood, we have two different options. One is low blood glucose and other one is high blood glucose. In both the cases, they will provide signal to the pancreas. Now, this pancreas is going to release hormones. In the case of low blood glucose, the pancreas will release glucagon by alpha cells, and that will trigger the liver cells to convert the glycogen into glucose. On the other hand, if you have high blood glucose, then pancreas will release insulin by beta cells, and this insulin will order the glucose to take up by the fat cells, and this way they will provide balance to the blood glucose level. In glycogenolysis mechanism, the mainly two enzymes are involved. They are glycogen phosphorylase and debranching enzyme. Their aim is to break the glycogen and convert it into glucose. Step 1. Glycogen phosphorylase. Glycogen phosphorylase removes glucose as glucose 1-phosphate from the glycogen. It contains pyridoxal phosphate, which is known as PLP, as a prosthetic group. The alpha 1,4 linkages in the glycogen are cleaved. It removes glucose units one at a time. Enzyme sequentially hydrolyzes alpha 1,4 glycosidic linkages till it reaches a glucose residue 3 to 4 glucose units away from the branch point. It cannot attack the 1,6 linkage at a branch point. If glycogen phosphorylase alone act on a glycogen molecule, the final product is highly branched molecule, so it is called as limit dextrin. Step 2. Debranching by bifunctional enzyme. Then a block of 3 glucose residues, it is trisaccharide unit, are transferred from the branching point to another branch. This enzyme is called as alpha 1,4 to alpha 1,4 glucan transferase. Now the branch point is free. Then alpha 1,6 glucosidase, it is a debranching enzyme. It can hydrolyze the remaining glucosal unit held in alpha 1,6 linkage at the branch point. This glucose residue is released as a free glucose. At this stage, the ratio glucose 1-phosphate to free glucose is about 8:1. The transferase and alpha 1,6 glucosidase will together convert the branch point to a linear one. With the removal of the branch point, phosphorylase enzyme can proceed with its action. Step 3. Phosphoglucomutase. Phosphorylase reaction produces glucose 1-phosphate while the branching enzyme releases glucose. The glucose 1-phosphate is converted to glucose 6-phosphate by phosphoglucomutase. In step 4, the glycogen phosphorylase further acts. Then glucose 1-phosphate units are released sequentially. The free glucose is released to the bloodstream. In muscle, it lacks glucose 6-phosphatase. It is important to remember that muscle will not release glucose to the bloodstream because muscle tissue does not contain glucose 6-phosphatase. Instead, in muscle, glucose 6-phosphate undergoes glycolysis to produce ATP for muscle contraction. In liver, the glycogen, which is converted to glucose 1-phosphate is converted to glucose 6-phosphate. Next, the hepatic glucose 6-phosphatase hydrolyzes glucose 6-phosphate to glucose. The free glucose is released to the bloodstream.
Advantages and disadvantages of glycogenolysis. Advantages: glycogen gives rapid energy source, anaerobically generated, 3 ATP generated per glucose from glycogen, 2 ATP per non-glycogen glucose. In muscle, it is a rapid source of energy. In liver, it maintains blood glucose levels. Disadvantages: low ATP and mass and limited storage.
Thank you for watching this video.